All right. Thank you, Lynn. Thanks for everyone that is still online. We started the next session. We will talk about fascinating ribosomal RNA modification, how to quantify it, how to map, and what is their biological role. So the first speaker is Yuri Motorin, Professor of Molecular Biology, University of Lorraine. Yuri, your pod is yours.
Okay, thank you. So first of all, I would like to thank Sami and Kate for this wonderful meeting and great program. So my talk is ribosomal RNA modification session, but many methods we are developing also applies to tRNA modification. So this is some kind of overlap. Okay, so traditionally, RNA modifications can be detected by different ways. So we already heard about different mass spectrometry coupled to TLC, and by all methods like RT stops at low DNTP was developed already in the 80s for detection of 2' methylation. But all these methods are not yet high throughput, even though there are a lot of progress in MS like we saw in the previous session. So we took another road and started to develop NGS-based approaches already 10 years ago in very tight collaboration with Mark Hell in mice. So basically after RNA extraction, RNA is subjected to chemical treatment and then to appropriate library preparation. We mostly use this small RNA kit with 2 ligations, but it's not in all methods, and the sequencing and of course bioinformatic analysis, which allows to extract the data. So since I'm supposed to talk mostly about ribosomal RNA modifications, so list of those is relatively limited. Even some other modified residues were reported in different other species. I put here only major species which were mapped and extensively analyzed. So bacteria have a great diversity of RNA modifications in ribosomal RNA. Many of those are not present in eukaryotic or probably in archaea, even though for archaea we don't know much, only few species have been analyzed in details. So how to approach detection of those modifications in practice. So for most of them, fortunately, we already have methods allowing their detection. So for 2' methylations, of course, we can use RiboMed-seq we are using, or 2' O-methyl-seq or NM-seq and some other methods even. So some others can be detected by like pseudouridine and some other modified uridines by hydroxy-seq or CMCT-derived methods or recently released BitSeq. So bisulfite is good for methylation of M5C, hydroxymethylation, and also methylation of position 4. And others are detectable either by RT signatures, many of them, because many of those nucleotides are RT arresting and generate visible signature in RT profile, or like Schrage developed a method for acetyl-4C, which can also be used. We were working on RiboMed-seq, alkaneamine-seq, and hydroxy-seq, allowing detection of some important modified residues in ribosomal RNA and also in tRNA, because MSLNG and dehydrouridines are also present. So I will not talk about antibody-based enrichment, because these methods are starting to be outdated now and replaced progressively, as we saw already yesterday, by more performing and better methods. But 2 other approaches are still extensively used, like direct chemical signature, which can be generated by modified nucleotides, which will introduce either mutations or abortive cDNA sequences synthesis during RT reaction. Those fragments can be captured and converted to library for establishing of this kind of profile, where we have a drop in coverage and sometimes or frequently methyl corporation at the position of a modified nucleotide. But those methods suffer from lack of controls because these profiles may be generated by other events in reverse transcription. So usage of specific chemicals or even enzymes, like we also saw yesterday, can help in distinguishing real RNA modifications from something else. And they are based on specific chemical treatment, which allow to introduce bulky residue arresting reverse transcription at a given point, which also can also be detected. So we are using, we started to use these RT signatures for detection of RNA modifications, and RT signatures may be relatively simple, like was shown for inosine, because inosine is base pairing with C, and of course, after sequencing, you have a G's appearing in the sequencing profile, or more complex, where there is a combination of read-through cDNA with mutation and abortive cDNA sequences, synthesis, which gives methyl corporation profile. So, of course, those modified nucleotides are generally modified at the Watson-Crick age, like you see here for M1A and M22G. So we worked together with Mark Helm on analysis of M1A modification using this type of profiling. So again, combination of reverse transcription arrest, methyl corporation, and correct read-through gives visible RT signatures in ribosomal RNA, here shown as example, 2 M1A sites in yeast ribosomal RNA, and of course, this signature disappears upon deletion of the respective enzyme, 1, 2, and both at the same time. So ROC curve, which allow identification of modification, is not bad. So the best AUC area under the curve is approaching 0.95, but it's still very, very poor for serious transcriptome-wide analysis of modification, because at this level of AUC, you still are having a lot of false positives. So the method allows also quantification of relative quantification of M1A, and by using of oligonucleotides with a variable sequence around, we found that sequencing signature is also depending on the neighboring nucleotides in the RNA. So as extension of this, again, in collaboration with Mark, we studied several different RTs, which differ quite considerably in arrest rate and mismatch rate, and also in jump rate, which is base skipping during the reverse transcription. And this reverse transcription signature can be also modulated by reaction conditions here, by variable concentration of magnesium, manganese ions in addition to magnesium. And all this is done with a goal to find combination of suitable RT and reaction conditions to achieve at least AUC 999 or even more, if possible, to get reliable data for large data sets. So in terms of chemically induced RT signatures, like illustrated here, we were developing and applying now quite intensively 3 major methods. So RiboMed-seq, which allows detection of 2' methylation, and of course a lot of projects are asking for detection and specifically quantification of 2' methylations in different diseases like cancer and others. So our probably best sensitive method is alkanin-seq, which allow to the detection of different modified nucleotides. I will talk a little bit more about them. And more recently, hydroxy-seq, which allow detection and, more importantly, quantification of pseudouridine in RNA. So for 2' methylation, there are different methods available on the market. So it can be detected by low DNTP-RT extension, so 2' or methyl-seq, by alkaline ribomet-seq we are using, or with periodate oxy-seq or NM-seq also introduced. So they use different principles known for decades now for all of them, like RT arrested low DNTP, exclusion of some fragments during alkaline hydrolysis, or resistance of methylated 3' end of RNA to periodate oxidation. So I will not discuss in details all these methods. All of them give a lot of false positives, and we are using alkaline ribomet-seq because, to our opinion, it gives the best quantification results over others. So I will talk more in details about alkaline-seq for detection of M7G, M3C, dihydrouridine, and also, as we know now, oxidized hydroxy-C and hydroxy-U in RNA. So idea, as was presented already by Richard Gregory in the previous session, comes from a very old publication from 70s and 80s, showing that M7G is sensitive to reduction by sodium borhydride. And we started development of this method by using this concept. After reduction, we have abortive cDNA synthesis, and these can be detected by sequencing. So we applied indeed this method with coupling sodium borhydride and aniline cleavage, but experimental results obtained with this protocol was relatively poor. Indeed, you can see here in the profile for wild type ribosomal RNA having single M7G residue that the signal indeed disappears in delta BAT 23 cells, which don't have modification, or when there is no treatment applied. But you can appreciate that there are a lot of false positives hits everywhere, and variation in those hits will really affect precision and specificity. So detection is possible, but specificity is relatively low. So we inspected once more the chemistry of cleavage and focused our attention to this particular phosphate, which appears after cleavage by aniline, which proceeds by beta elimination. Chemistry is very well known for DNA and also for RNA. So, and we sought to use this phosphate for specific ligation to somehow enrich fragments starting from N+1 nucleotide resulting from the cleavage. We also accidentally found by analyzing different controls that just alkaline hydrolysis without borhydride reduction is sufficient to pick out M7G and also some other modified nucleotides I will talk in a moment. So our procedure consists in alkaline hydrolysis, which will fragilize M7G and some other modifications, then dephosphorylation to remove all possible phosphates which may give a background, and aniline cleavage, which reveal this phosphate and N+1 nucleotide. Then we apply standard methods for adapter ligation at the 3' end and 5' end and amplification and sequencing, resulting in this type of alignment of reads at N+1 nucleotide in the sequence. Then we have to score the sequence, the signal with different scores indicated here. So as a result, the method is really very, very sensitive, because when we made this mix of ribosomal RNA from BAT23 and wild type strain. So but BAT23 has no M7G in 18S ribosomal RNA, which was shown here. So you can see that the signal is still visible until this point, which is 2.5% of molar ratio of M7G in tRNA mix. Then it roughly in the background, so we cannot quantify it at the low proportion. So as you see here, calibration curve is non-linear for both stop ratio and normalized cleavage. So we are very, very sensitive for low percentage of modification, but I would say a resolution of precision for quantification is lower when molar ratio is between 50 and 100%. But sensitivity is very high. And signal is dependent really on the presence of M7G and aniline treatment. Without aniline or in deleted strain, there is absolutely no signal at this position. And also for 25S ribosomal RNA, the ground is very, very low. So we found that the methods can also detect M3C, here shown for position 32. And we still don't know what is the chemistry behind of this, because M3C at the nucleoside level just deaminates to M3U, and M3U is not detectable by the method, but something else happens in RNA chain. And not surprisingly, dehydrouridine is cleaved upon alkaline hydrolysis, opening in this uridopropionic acid, and which can be kicked out by aniline giving the signal. And we also found that something happens with hydroxy 5C, which is rare modified nucleotide in bacterial ribosomal RNA, and also with hydroxy 5U, which is intermediate in the biosynthesis of complex modification in tRNA anticodoning. So detection is very sensitive, but quantification may be a bit complicated because there is saturation of calibration curve. So, and the last method I wanted to show you in application for ribosomal RNA is the most recent hydroxy-seq protocol. So we thought that CMCT-based detection is not really perfect for quantification, and we used also very, very old chemistry reported already in 70s and beginning of 80s, where hydrazine and Richard Ruggero also talked about this, but we use it in a slightly different conditions. So hydrazine cleaves uridines rather efficiently, but pseudouridines are resistant to hydrazine cleavage at these conditions. So, and their reactivity is roughly identical to A, C, and G nucleotides here. So chemistry is shown here. So chemistry is very, very similar to the one we saw for alkanilin-seq, but concept is more related to ribomed-seq, because we are cleaving all RNA molecules at uridine residues, but not Psi. So Psi are protected from cleavage. So there is a dephosphorylation step with T4PNK to remove only 3' phosphates here and not 5' phosphates, because we need them for adapter ligation. And after adapter ligation sequencing and mapping, we have this type of profile. So all reads are aligned to N+1 nucleotide to U, and pseudouridines should be relatively resistant for that. So this method can be used for both detection and quantification. And this is shown here as example for 5S ribosomal RNA. So it's a log scale, so that's why we have a variable background, which is used for normalization in this gray color. And the signals after normalization looks like this. So we have cleavages at all U's represented by T residue in the sequence, and position of pseudouridine 50, which is fully modified or almost fully modified in ribosome RNA, is well protected from cleavage. There is no cleavage at all at this particular position. We also found that the same chemistry can detect M5U, because M5U are also ribotimidine is also resistant, and some other derivatives of modified U at position 5. So those are protected, and lysidine, so K2C in tRNA, is cleaved, very efficiently cleaved. This also allows detection and quantification for this reason. So I will just talk a little bit about validation problem, which is major problem in the field. So of course, it would be great to have a very high intrinsic specificity of the method. Of course, when you have this type of profile, it's very difficult to validate something, because a lot of background and a lot of parasite signals which are not resulting from modification. Of course, we would prefer this type of picture, but it's not always possible. Here we have really very good enrichment of N+1 nucleotide to modification, which is not always the case. So another idea, which is more and more implemented in the field, and this is a very good sign, that we can use orthogonal specific approaches to confirm that the signal we see, for example, in ribomet-seq, corroborate well with a low DNTP-seq or hydroxy-seq with CMCT-seq. And of course, this can also be done in combination with LCMS, or now with nanopore sequencing, both in direct RNA sequencing and cDNA sequencing also. So a very good method, which I would recommend to everybody, of course, the usage of KO deletion mutants when it's available, because when you have a signal corresponding here for modified nucleotide in tRNA, and in KO it disappears, this very good sign that indeed the signal corresponds to RNA modification. We also recommend more or less a usage of synthetic RNA transcripts, T7 transcripts, but with some limitations, because sometimes, like you see here, cleavage may be slightly different between in vivo modified RNA and in vitro T7 transcript, and this makes interpretation of difference quite complicated. So, of course, analysis of biological triplicates is...
Sorry, we are 3 minutes over 20.
Yeah, I'm almost there.
Oh.
So, analysis of in biological triplicates, duplicate is a strict minimum, is good, but imagine you have this, you are happy, but what you will do with this type of profile, even with 3 samples, it's very difficult to compute something. So since modifications can be variable under experimental conditions, they may be modulated by different parameters. So control of experimental conditions should be very, very tight. And we need, of course, quite substantial coverage to detect modification in practice. And what was not discussed really a lot for tRNAs, but for many RNAs, we need non-redundant and reliable reference sequence to map the reads on them. Okay, so I will skip this slide, and I will thank my collaborators for this work, which is done in Nancy, so members of the team and sequencing core facility, and members of Mark Helm Lab in Mainz. And thank you for your attention.
Great. Thanks, Yuri. So let's see if we have any question. I don't see any question at the moment. However, I'll ask a question, and maybe we can also discuss maybe more in the panel. This is very exciting, new technology, maybe more to understand how your sequencing approach will compare with obviously other technique, right? To map or quantify modification. It might be a long answer, so if you want, you can.
Right. I think it would be great to go to panel session for this. So I will share.
Great.
Stop sharing my screen.
Thank you so much, Yuri. Thank you. Yeah, expression of the host gene. Yes, always a very, very important point.
That's great. Yeah, good question. Well, thank you again. Just as phenomenal work.
Thank you.
I'll turn it over to you because I know you're going to introduce our next speaker, but really fantastic.
Thank you. Yes. So our next speaker is Sarath Janga, and I believe he's an associate professor Indiana University. Take it away.
Thank you, Davide. Can you see that all right?
Yes, I think it's going to be fine. Yes.
All right. So yes, I am Sarath Janga. I'm an associate professor of bioinformatics and data science at the Luddy School of Informatics, Computing and Engineering at Indiana University. First of all, I would like to thank the organizers, Kate and Sammy, for inviting me and hosting this fantastic virtual symposium. So as the title suggests, what I thought I'll do today is give an overview of some of the work that we have been doing over the years using single molecule direct RNA sequencing for mapping RNA modifications and to also resolve secondary structures of RNA. So a lot of the work is going to be more on the foundations that we have set, and where possible, I will also be updating on our most recent updates or outcomes of this work. So let's see. So the field of RNA modifications does not need much introduction to this field, to this group of audience. RNA modifications are broadly defined as the chemical alterations of regular nucleotides of RNA. For instance, adenosine to inosine conversion is facilitated by ADAR class of enzymes. Uridine to pseudouridine is facilitated by the pseudouridine synthesis. And several folks in the symposium have talked about M6A, M1A, 5-methylcytosine, and so on and so forth. So we now know that there are about more than 160 modifications that occur across a broad set of RNA molecules. However, most of the work that has been done has been largely done to profile modifications for or sites of modification for M6A, A2I, pseudouridine, and M5C. And you'll see in a few minutes why I stress that most work has been focusing largely on this, because what we know can only help development of some of the machine learning models that we are interested in. So other aspect of RNA modification that is very fascinating to me is the classification and their occurrence on the RNA molecule. So there are many ways you can define RNA modifications. One way you could do that is based on base editing. For instance, A2I facilitated by ADARs or CTU facilitated by ApoBex can be called as base editing modifications, while the chemical alterations, which basically do not necessarily involve a base change, such as 5-methylcytosine, M6A, M1A, and so on, or even the recent interest of a lot of groups, the 2' methylation, the backbone modification. These can be considered the chemical alterations. Now, what is exciting to the field, at least from my point of view, is RNA molecules can have one or more of these modifications occur in combination. And I'm trying to look for the pointer. So when you look at a single molecule of RNA, you could see that one or more modifications can occur along the length of the RNA molecule, and these could give different functional and phenotypic outcomes as the cell progresses. And however, we are far from clear on how this combination of modifications could occur and how these would drive the RNA isoforms as the cell progresses. So that is something I think is an open-ended question for the community. Another aspect of this RNA modification world is the concept of epitranscriptome, which has been heavily appreciated in the community for the past decade or so. This basically relates to the notion that RNA can be modified by the writer class of enzymes, while the erasers end up removing these modifications, and the readers sit on this modified RNA to facilitate different functional outcomes. For instance, if you focus on an M6A encoded mRNA, you could have the writer class of metal class of enzymes, which would install the modification, while the FTO or ALK-BH5 kind of erasers would remove it. While the readers, several RNA binding proteins which can specifically recognize upon modification, could result in the functional outcomes in various forms, such as translation, splicing, and nuclear export. While a lot of this machinery is well known for M6A, our understanding of how this compendium of this broader class of RNA binding proteins are shaping the other modifications is largely unknown. Again, this is a work in progress for several groups. So we have had some successes and progress, but still the compendium is far from complete for several modifications. Again, from a mapping perspective for this modification, currently the traditional methodologies, again, other colleagues have talked about some newer methods that have emerged, but traditionally, most of this mapping has been done using either antibody-based or a chemical-based modification. In antibody-based methods, you'd use an antibody and pull down the target and end up doing a short read sequencing, which often results in not necessarily accurate nucleotide resolution mapping, but a general localization of the modification site. The chemical-based methods often end up giving a bit more nucleotide resolution of the location of modification. For instance, bisulfite sequencing is commonly used for M5C, and sodium bisulfite is the reagent that I've highlighted in this particular case. You would typically see when you do this, you would have the regular cytosines are modified while the M5C is unmodified, resulting in the short read data having changes for cytosines, but not for the M5C. Now, what we know from several studies is that there are relatively fewer techniques for these modifications, and most of these techniques are unique to each modification. So they would detect only one type of modification in a given sample. They would often have a higher error rate or low resolution. And again, Yuri and other colleagues have pointed out to these challenges. And in any case, each of these are available for one or at most, sometimes a couple of modifications with slight modifications. So if you wanted to map these modifications on a cell line, you would have to use multiple methods to map the bunch of modifications. So seeing this, years ago, we developed an in-house database called Epitome, which currently houses about 3 million sites for about a dozen modifications in human and mouse. You would be able to look at a particular gene or a modification type or based on coordinates, and it would show the series of locations where the modification happens along with the study and where possible the tissue-specific expression for this particular gene of interest. So this was developed largely as an in-house resource to facilitate our machine learning AI models, as I will be alluding to in the following slides. So in the lab, one of the first method that we developed was for predicting M5C. It's called Raven. We are very fond of birds. So M5C, we have called it Raven, and for pseudouridine, we call it Penguin. But then the postdoc didn't like these bird names anymore, so she started calling different names. For NM, she called NM Nano, and so on. So, but anyway, the idea behind these methods is to use long-read direct RNA sequencing from nanopore. So before I get into the details of how these methods are developed and improved over the time, in nanopore sequencing, when a single molecule of DNA or RNA flows through the pore, tethered to the motor protein, there is a characteristic disruption of current, as you can see in here. And this disruption of current often for a direct RNA sequencing corresponds to about 5 nucleotides that are present in the nanopore. So this is a continuum of current versus time axis. So as the molecule flows, there is an overlapping set of current signatures corresponding to different fimers that are. And also of interest in this context is in a typical nanopore flow cell, there are at least 1,600 pores, which are simultaneously sequencing several RNA molecules, resulting in a large amount of data and also need for GPU accelerated for live-based calling. And thanks to parallel improvements in machine learning as well as GPU resources in the past decade, nanopore has had great success in coordinating these into a single compute and GPU modules for live-based calling. From an experimental point of view, so far, nanopore has been able to do direct RNA sequencing on full-length mRNA molecules. And this is facilitated using typically an oligo-DT primer, followed by a reverse transcription step, which we often end up using. And we do that because it stabilizes the RNA so that there are no secondary structures formed. And the subsequent step of attachment of 1D sequencing adapters, tethering to the motor protein, this whole process takes about a couple of hours. So it's fairly easy to generate these libraries. And we have done it for a number of cell lines, generating about 1 million reads or more for each independent run of each independent run. We have also had limited success on primary tissues such as cortex, cerebellum, and hippocampus, which I'm not going to go in more detail because the focus today is largely on modeling part. So when we look at these kind of data sets, these charts, histograms show the read length on X and the number of reads on the Y-axis for a liver cell line and a leukemic cell line. As you can see, these are truncated at 4,000 base pairs. And you can quickly visualize that there are still differences in the transcript lengths between these two cell lines. However, they do capture essentially for a large number of genes and transcripts end to end. So, as I said, the premise for nanopore direct RNA sequencing to facilitate modification detection is that when a molecule flows through the pore, there's a current signal that is generated. And this current signal is believed to be different for regular versus modified signature. And if you notice here, I've marked XXC, XXS. So this is defined as a k-mer or a phi-mer, where a phi nucleotide signature is a focus and the current. When you look at this, it's not easy to distinguish what exactly is the current versus time. But when you actually look at a large sample of signals, you can actually distinguish. And again, as you will see in the next set of slides. So our goal is to generate a 256 k-mer where the middle base is fixed and a modified nucleotide corresponding signature is also generated for current, and then use it for machine learning and AI techniques. On the top to the right, you can see an example of a current versus position for one particular molecule. This was from the Oxford Nanopore Technologies, one of their first studies for direct RNA sequencing. You can see that the unmodified position has a higher current signature compared to an M6A M6A position at the same point. So there is a difference, but you need a good chunk of a sample to actually start modeling. So one of the tools that we developed is called Sequoia for visualization of signal features where current differentials can be identified. And other groups have generated tools like Tombow and Nanomod to essentially serve the same purpose of visualizing these current signals. So one of our first efforts was to see whether we can develop these modification models for prediction of methyl cytosine. So thanks to our rescue, there are a few groups which have generated bisulfite sequencing for HeLa cell line. So what we did was collection of this bisulfite sequencing data for identification of M5C. And you can see from the histogram to the right, there are a small number of modification sites. There are a large number of modification sites which have a very low probability of modification. So by probability of modification, what I mean is if at a particular position there are 2 reads which are modified out of 10 reads that are mapped, that gives you a probability of 0.2. Other groups have been calling it as editing levels or modification levels. And you can see from this histogram, there is a small but a significant number of sites which have a probability of 0.6 for modification extent. So as you will see in the next slide why this probability of modification becomes important for modeling purposes. So all this data that we took was from studies which are reported and readily available from our in-house resource, which is publicly available. So we performed a HeLa cell direct RNA sequencing and extracted the signals corresponding to these roughly 2,500 locations with varying modification probabilities. And extracted the signals corresponding to about 20,000 signals, performed training and validation using machine learning and deep neural network models. So, first thing that we did was just to check whether the modified median current signals for the modified phi-mers versus the unmodified is actually different. You can see that the current signals are actually different, and we checked for various other parameters, and we do see variation. So this resulted in trying a number of different machine learning and deep learning models at various probability levels. For instance, at 0.6, not surprisingly, where 6 out of 10 reads at that location are actually modified, you end up seeing higher success. You end up seeing higher probability. And since this original work, we have seen that increasing the amount of data from various groups improves the success for our models. And for instance, for pseudo-Uridine, using pseudo-seq data, we have recently published a paper showing that there is a 92% accuracy training on one cell line and predicting completely on a totally new cell line with these sort of models. So what are the challenges with these kind of models? Main challenge is the false positives in the gold standard so-called Illumina-based data sets, or the amount of data being less, because if you don't have a good number of combinations of these 256 MERS, you don't have a complete capture, or the experimental approaches may not be available for most of the modifications, limiting. So there, it is in this context, we and others have been using synthetic RNAs embedded with RNA modifications where possible, but that is also a challenge, which is a topic of a different discussion. Yet another area where we have been using nanopore direct RNA sequencing is to map the secondary structure of RNA. Again, to this audience, I don't need to do much introduction to the structure, RNA structure. RNA structure is dynamic, it responds to signals, and is known that the secondary and tertiary structural elements can be interconverted depending on the presence of proteins, electrolytes, and small molecules. So, however, most of the work of our RNA structure for many years has been to largely determine the 3D structure, which are time-consuming and are limited to specific RNAs which exhibit fairly stable structures using X-ray and NMR techniques. In the last decade or for longer, people have been spending time to do secondary structure mapping using high throughput sequencing. These involve a range of techniques such as ShapeSec, DMS, IC-Shape, and LiGeR-Seq, Paris, and so on. Now, the premise of all these methods is that when you have an RNA molecule in solution, if you use dimethyl sulfate or 1M7, which is a ShapeSec reagent, these reagents would specifically modify the single stranded region in a kind of a random manner, but would not affect the double stranded regions. And you can see these moieties that are attached because of a treatment. Now, when you subject these to reverse transcription, there will be a dropout of the reads at these modifications. Thereby, the sequencing output would enable you to identify wherever these modifications are more abundant. For instance, in locations where the single stranded regions are modified, you will see more dropout and you will see higher reactivity score represented as the number of reads or some normalization of thereof. So again, the approach in these is you take an RNA molecule, you treat with one of the shape or DMS reagent. On the left, you would follow the using a reverse transcription and short read sequencing. On the right, you would, for instance, do a control using DMSO, and you would get a control sequencing data which would not have peaks because of the drop-off from the reverse transcription on modified locations. And because of accumulation of reads, you would have higher reactivity scores in locations where there is unstructuredness and lower reactivity scores in structured regions. Now, most of these protocols have been using short read approaches. The challenge being that when you use short read approaches, you cannot have an isoform resolution, and you would also not be able to deconvolute if a single RNA molecule exhibits an ensemble of structures. So we thought, how would we explore using long read direct RNA sequencing on a shape-treated kind of a library to see whether these modified locations can be captured either using SNPs or from base calling of the nanopore data, or by directly developing a model for these modified locations. So the idea here, so we used a pilot sample of an RNA molecule which is about 338 nucleotides long, which is a Hep C virus internal ribosome entry site, and performed a shape treatment, which is an 1M7 reagent. As you can see, this moiety is fairly bulky. So we would end up seeing either sometimes blockage of pores or definitely seeing change in the current. We also did a polyadenylation followed by ligation, flow cell, priming flow cell, and direct RNA sequencing. So broadly speaking, you would see that there are 3 steps: a synthetic biology step where you take this RNA molecule, perform treatment with a shape or a DMS or other reagent. You would also have a controlled molecule, which would then be polyethylinated, and you would perform both these libraries, you would do a direct RNA sequencing of both these libraries, with the idea that in the library which has modifications that would be captured, while the end product is that you would align these reads, and wherever there would be SNPs, that would help you to identify, generate a reactivity score at that particular location, and you'd use the control for finding the differential. So our computation pipeline involved filtering of reads which are at least having a 70% alignment, because we did see some fragmentation and some reads which do not completely map and so on. So we filtered for reads which had a good extent of alignment. Performed in initial approach, we just identified the SNPs using blast to SNP, calculate the probability of modification, generated what are called as reactivity scores, and we generated a bunch of reactivity scores, as you will see in the bottom part of this slide. And these inferred reactivity scores are then fed into RNA structure or other structure prediction algorithms as a constraint. So the reactivity scores can be generated using normalized modification probability in the treated library compared to a control library. And you can either do X minus Y at each position, or just take the modification data, or take a logarithm or various kind of metrics. From our approach, we noticed that, again, you will see on the top, the y-axis is extent of SNPs that are seen at a particular location. The y-axis is the extent of modification, while the x-axis is the position, nucleotide position. So the shape-treated control, and the bottom you see the difference, which is the log of x by y. We noticed that in most of these locations where a short read data has been already used for the same molecule, we're using to generate the structure, we had an agreement with the scores. So we ended up using log of x by y as a reactivity score. And you can see from here, when these reactivity scores are used as input for RNA structure, the leftmost is the inferred structure from the long read data. The rightmost is a structure available from EBI. The middle is the reported structure using short read data from Julius Lux and other colleagues using this RNA molecule. So we compared the structure for end-to-end, and we—
Sure, Sarah, that we have 2 minutes more. Okay.
Oh, I'm almost done. This is the last slide.
Thank you.
So we see a pretty good agreement between the inferred structure and the reported structure so far, which enabled us to suggest that indeed long-read direct RNA sequencing can be employed for end-to-end mapping of structures if you do a deep sequencing one. Second, if you use appropriate controls. And we have since then generated a library of software that is work in progress for identifying these modified nucleotides from shape-treated. So with that, I would like to thank Julius and Molly for early help in developing this structure probing work. And the work on RNA modifications has been done by several members in the lab over the years. Originally, it was done by Kosinamir and Raja, and more recently, Doa took over along with a couple of grad students to start mapping these modification models. With that, I'll stop here.
Great. Thank you so much. This was really very great talk, very cool technology. Let's see if we see any question, although we have maybe one question. One thing maybe again would be maybe interesting maybe for the panel, to based on your technology related to try to combine how modifications, right, of RNA and it can actually change obviously structure, right, using your technology, right? So that I was intrigued when you were talking about this, right, to combine the two things, right? Yeah.
So yes, I think our long-term interest is to not only map modifications, but if you can do it in the same cell line or same sample, parallelly the structure, you can start studying structure and RNA modification interplay.
Exactly. Anyway, great. So let's continue this discussion on the panel. But it was great. So next speaker, yes. So Cynthia Burrows is a distinguished professor of chemistry at University of Utah, and I believe you are sharing the screen. All right, Cynthia, take it away.
Let's see. Okay. Hopefully
Yes.
you're seeing the full screen. Thank you. I'm sorry, my title just kept getting longer and longer. Last night I added bisulfite sequencing. So this morning I thought, okay, let's make a short title. We'll just say we're trying to sequence everything everywhere all at once. So how are we going to do that? We picked the E. coli ribosome as a place to start because it's well known, the modifications are well established, and the writer knockouts are readily available. In the E. coli ribosome, we've got 17 different structures at 36 different sites. And so we thought we would try to map all of these with direct RNA sequencing with the nanopore. Now, before I go on, I need to tell you about disclosures and give some credit, and especially I need to give credit to Aaron Fleming, who's a research associate professor in the lab, and he's really the master of the nanopore. So everything I talk about, he's had his hands on, as well as other members of the group. Okay, so we're going to use the ONT system that you've seen now many times before in the last 2 days. And green and black is the nanopore protein with a constriction zone in the middle, and this constriction zone that has about 5 nucleotides in it contributing to the electrical signature, as you see here. That sensing zone is also called the K-mer, as you've heard in previous talks. Now, there's another protein, and that's the helicase, which is sitting on top, and the helicase is regulating the speed at which the RNA is being delivered to the nanopore. And this helicase operates as an ATP-driven motor, so the concentration of ATP and its hydrolysis determines the rate at which DNA or RNA translocates through the channel. In our work, we principally use Eligos 2 and nanopore SU. The latter is from Tal Pan's lab. Eligos is from a group at Arkansas Med School. And I'll be talking exclusively about the version 9.4 flow cell chemistry today. So of course, the question is, how does the system respond to modifications? And before telling you about our work on the E. coli ribosome, I want to tell you a little bit about the SARS-CoV-2 viral RNA. We heard about this yesterday from the group in Italy, and I just want to reiterate a few things because this is where the dwell time signature really came from that has been so useful to our lab. So you heard already about the subgenomes that are mostly the 3' end of this viral RNA. And what we reported a couple of years ago are the 5 high confidence sites, as we called them then, of the 3' end that are pseudo-uridynylated at the sites that you see here. Later, we were able to put some numbers on these. I'm not sure that 2 significant figures is correct here, but at least one probably is. And these were quantified by using nanopore SU. And so, and we verified that these were written by +1 and +7 into the SARS viral RNA. So how did we figure out that these were well-established pseudouridines? It was first of all by looking at base calling. So if you have a long RNA that's either U in all of its native locations or pseudouridine, in the first experiment, when you have U in many different sequence contexts, that will be base called mostly as a U with pretty good accuracy. A couple of sequences are not quite base calling correctly, but it's pretty good. When you have pseudouridine, even at 100% occupancy, it might call either as a U or as a C. So if you look on the right-hand side, there are sequence contexts where pseudouridine will be called as a C. And in fact, partway in between, it looks like a mixture of C and U. And so we need to have standards with all these different sequence contexts in order to be able to figure out what amount of pseudouridine do we really have. So that's done by synthesizing RNA by in vitro transcription, either using uridine triphosphate or pseudouridine triphosphate, or by synthesizing short oligos that contain U and pseudo-U in appropriate places. We make those by solid phase synthesis of ligate into longer RNAs. Okay, so from there, we can then look at current levels, and the current for U or pseudo-U and the base calling will give us some clue as to what is going on with the at that particular site. But a key thing that Aaron Fleming noticed was that the other protein is also reading the base. And I say that because it is, the helicase is responsible for pulling apart a base pair before it goes into the nanopore. And depending on how well stacked that nucleotide is, how well stacked that base is, then it'll be easier or harder to pull apart. And we found that with pseudouridine, it was slower to be processed by the helicase. Actually, it was a double Gaussian, as you see here, probably because of the syn and anti orientations possible for pseudouridine. At any case, that slowing of the helicase is something that we could measure. So again, base calling error is one thing that where you will get things moving high above the baseline. Current intensity is another place where we'll see things being jumping up above a baseline, a background level. But then to verify a pseudouridine at a given site, we look 10 nucleotides to the 3' side and see if there's a dwell time signature. And if there is, then this site, this U, was probably a pseudo-U. And so that helped us enormously to try to figure out which sites were really pseudouridine. And so why does this happen? Well, again, we think it is because the helicase has phenylalanine 350 stacked against the base pair that it's trying to pull apart. And if that pseudouridine is well stacked, then it's a little slower to pull apart. Actually, most modifications are easier to pull apart because the modification on the base usually is destabilizing to the base pair. But so pseudouridine is going in the opposite direction from those. I also want to point out that lysine 289 is here, and it looks like it interacts with the ribophosphate backbone about 2 nucleotides earlier in the process. I'll come back to that. That would be 12 nucleotides. It'd be higher up here, 12 nucleotides away from the constriction zone. So save that information for later. Okay, so let's get back to the ribosome. I skipped over this again, but we're going to use all three: base calling error, current level, intensity, and helicase dwell time. Those three are going to be the way that we look for each of these modifications throughout 16S and 23S ribosomal RNA. And so here are all the results. For those 36 locations, sometimes we saw a change by the base calling error that was introduced. Sometimes there's a change in current level that can be analyzed by, for example, by NanoCom4 or other programs. And sometimes there's a change in helicase dwell time. Sometimes we get 2 out of 3. And I have to say the most difficult for us was 5-methyl-C. That's the one site that gives only a tiny, tiny change. So for these, we also studied synthetic RNA. When we had the triphosphate available, we could insert that into that Kmer synthesized strands and look at various Kmers. Then we made writer knockouts, grew those up and isolated E. coli and looked at those as well. And so for now, having all of these mapped in terms of what modifications really look like in the E. coli ribosome, and remember that these are all written at fairly high levels, usually greater than 90%, we were then set up to look at stress. And we used stress conditions that had been reported before in the literature: cold shock, heat shock, acid stress, metabolic stress, etc. And sometimes the E. coli do not grow so well under these stresses. At any rate, we could isolate enough RNA to study it, and then we can look to see where were the modifications reduced. For the most part, they're reduced because they're written in at such a high level in the first place that they are then reduced under stress. Although here's an example, the green one up top, that's an example of one that increases. That's a pseudouridine, actually. It increases under stress. If we look just at the pseudouridines by nanopore Su, that there are 2 of them that perhaps increase. Most of them decrease a bit or stay the same. And so if we then look at all 36 modifications, we can say whether they stay the same, that's in black, or change a little bit, that would be in yellow, or change a lot, that's in red. And the one that goes up is all the way at the bottom, that's an increase. Let's say 2604. Okay, so again, we analyze using a couple of different types of software: metabolic stress and cold shock. We also looked at heat shock, but the data is not quite as clean because when you do heat shock, RNA tends to cleave. So there's not as much data there. At any rate, you see that there are a number of positions that are changing, trying to adapt to stress. I'll just mention 2 of these that we thought were really interesting. We heard, I think it was Yuri told us this morning about 5-hydroxy-C. I think that's a really interesting modification because it's the only oxidized base in the ribosome. It's a really interesting biosynthetic pathway elaborated by Tom Suzuki as well. And another one that changes quite dramatically is the dimethyl-C at position 1402. So I'm going to tell you just about that for a moment. So the dimethyl-C has 2 different writers that install a methyl group on N4 of the base, and on 2' is a different writer. And so you can obviously tell by mass spec that one of these is going away, but the question is which methyl group is going away. And actually we could figure this out by the signature from the dwell time. So on the bottom, I show you the native E. coli grown at 37 degrees. and the position of interest is 1402, but for dwell time, we'll look 10 nucleotides toward the 3' side, and we see there's a dwell time signature there. That's interesting. There's also another change that occurs at 12 nucleotides. And so this double signature, we would like to think is due to the 2 different methyl groups, one on the base possibly being impacted by that phenylalanine that pulls base pairs apart, and the other one maybe is being impacted 2 nucleotides earlier by another residue that contacts the backbone. At any rate, there are 2 signatures here. Under metabolic stress and cold shock, the first 10 nucleotide signature goes away and the other remains, and it also remains in a knockout that takes out that does not write then the base methylation. And so that's telling us that actually under stress, we lose the methyl group on the, or we don't put in, I should say, the methyl group on the base, although it is retained here on the 2'-O-methyl. And so why is this? Actually, this site has been studied in the ribosome, and this is a point at which you see this dimethyl C is impacted by protein Y, and if that methyl group is not there on the base, then PY is released and it sets up the organism for hibernation. So it was very interesting to see that result, be able to monitor that. Okay, the other thing that we've seen is that the ribosome in E. coli has 7 different operons, and these operons are very nearly identical, but they have some sequence variation. I'll just point to one site here on the right, 93 in 16S RNA, and that site is U in some of the operons and C in others. And so if you think about it, if you sequence all of the ribosomal RNA together collectively, that site looks like a good candidate for pseudouridine, but it's not. It's a sequence variation. Those are legitimate C's and U's at the same site. And so thinking about this, we realized we should also be looking at modifications as a function of each operon. And also, we can, with the nanopore, simply count the different sequences and see how they change under stress. And so what we found was that under cold shock stress, certain operons, B, E, and H, to some extent G, are not being expressed as much as C, A, and D. On the other hand, under metabolic stress is a little bit different pattern. And this, we found a nice report from Scott Blanchard's group where they have studied this in a different way. We are very happy to say our work is consistent with what they published a few years back. Okay, and so from this interesting point about the CU signature, I want to just remind everybody about the chemistry that comes out of bisulfite sequencing. You remember that C is converted to U by bisulfite, and 5-methyl-C does not react. But we found in collaboration with Brad Cairns a few years ago that pseudouridine also reacts with bisulfite. It does that very cleanly. And in fact, if you do it at pH 7, only bisulfite reacts to form an adduct that gives a very clean signature in high yield. This has recently been further developed by Tuan He and by Chen-Chi Yi's lab, and their procedures are now published. And so we looked at whether or not we could make these bisulfite adducts on pseudouridine and then pass the adducts through the nanopore. We would expect, this is now so different from U and so different from C, we should get a big change in base calling error, and we do. So in the purple, the adducts are showing a huge error at the position X. And that gives us more dynamic range for looking at quantification of modification. At 10% modification, it not may be significant, but at 20%, we do see a difference between pseudouridine and the adduct. And so this is giving us more confidence that we could be more quantitative. And so we looked then at some of the known pseudouridine sites before treating with bisulfite is on the left. Running the bisulfite adduct through the nanopore, you can see that the signature changes a lot, especially U really goes away. For the 1911 position, also a big change, 2457, again, fairly big change. Whereas those sites that are CU variants, when you treat with bisulfite, nothing changes. And to show just one final example, I think this is my last slide, we looked at the tmRNA loop, T-loop, that is E. coli RNA. This is 363 nucleotide RNA that labels failed proteins. It had been studied years ago by some of my colleagues here at the University of Utah. And Psi 342, this one, was thought to be installed at greater than 90%. 347 was thought to be only about 10%. But this was according to CMC labeling, which is not the best way, in my opinion, to look for pseudouridine. It's also a highly modified loop. There's a 5-methyl U there. So if you walk through this whole loop, starting at this 5' G, bisulfite shows no change to the first G, almost no change to the next G, not much change at M5U. This pseudouridine changes a lot, then C, A, C don't change much. And this partial pseudouridine does change. We can guess its occupancy at about 25%. And the last two C's don't change. And so I think this combination of bisulfite-assisted nanopore sequencing should be very helpful as we go forward examining specific sites. Okay, so this is what I told you. We've looked at 17 modifications at 36 locations. We've used the dwell time and bisulfite adducts when necessary to enhance these signals and to give us enough information to make assignments. Finally, I thank these people again, and I thank you all for your kind attention.
Great, thank you so much, Cynthia. It was really great, and it's very exciting. The bisulfate assisted nanopore seems that we are at least we are moving forward in the great directions. One question, a quick question, maybe I missed this. During the stress or metabolic stress, the enzyme that modify the ribosomal RNA nucleotide also changed in expressions? Like how much they changed?
Wait a minute, tell me the
enzymes, right, that modify the specific ribosomal RNA nucleotides, right? When you stimulate the cells, when you stress the cells, right?
Yep.
How much do you see, that the enzyme will in the term of...
I don't think we've looked at changes in enzyme level. Is that known? Yes. They all go down? Yes, Aaron tells me they all go down. Thank you, Aaron. So the enzyme levels are going down. So presumably that's a large part of what's responsible for the writing going down. But of course, some of that could be, is it also going to be SNO RNAs? and their availability to write modifications. That could be important, and we haven't looked much at eukaryotic ribosomes, but that could be an important thing to look at next.
Yeah. Great. So there is a question from Luis Gracia Mazuka. How did you assess the level of expression for the 7 operons of coli ribosomes? Did you have to discard the nanopore reads that did not cover position that differ in the different operons?
Did not cover, but I mean, we get good reads. We can pretty much go through, you get excellent reads, especially at the 3' end of the sequences. And for at this point, we're getting up to a million reads, and you can just pool these data now, because we know the individual sequences of those 7 operons. You can just pool the data and count, so we know how many we have. Yeah.
All right, great. Are there other questions for the panel? Don't be shy. Okay, so maybe we can carry on this discussion during the panel session. Great. Well, thanks for everyone to be back. Yeah, so the panel definitely would like to discuss some of the questions that we were already raised during the talks. Actually, there is already a question that is a perfect segue. This is from Macy Slaughter. That's actually one of my students. It's perfect, similar to what Scott was saying, right? So if a junior scientist started to enter in this field, what is the best technique, right, to use to monitor and quantify ribosomal RNA modification? So Macy asked, for example, how does nanopore sequencing fit with the orthogonal approach mentioned, right? So do you need to do other approach if you use nanopore?
Well, I can take a stab at that. I think it depends on what your experiment is. So, personally, I think the nanopore is great for giving long reads and for high abundance modifications, and for monitoring sort of overall qualitative changes. If you want really highly precise data that something is changing from 72% occupancy to 58% occupancy, then you better go do mass spec, right? Because the nanopore is not going to be that accurate usually. But if you want an overall picture, like we were trying to look at, basically who goes up and down and what changes during stress, then that I thought was, that's a good nanopore experiment. I think mRNA lends itself, and viral RNA really lends itself to the nanopore method. But high density modifications or subtle modifications, like Pete Didon was talking about, these different prenylated intermediates along the way, I can't imagine whether or not the nanopore would pick those up.
Yeah.
For example.
I think it's a good point. So however, you were showing that you can detect even 20%, right, differences. And I think it's an important concept here to make that if we study ribosome, right, ribosomal RNA modifications, right, if we can detect even 10%, difference, considering at least there are millions of ribosomes, right? you can take. So I think the point is the 10% or 20% is still maybe extremely helpful, right, in terms of a biological impact, right?
Mm-hmm.
So I mean, of course, it's different if we study obviously and a very poor, like, I don't know, expressed transcript, right? I can see that. But I think in the context of the ribosome, there is a little bit more some difference, right? Compared to monitor other modification on other RNAs, yeah. Anyway, this is Cynthia, yeah, that is great. I don't know if Yuri or Sarath, you want to add on this point that I think is a critical actually in the field?
Yeah, so I think the way I look at it is the pros of nanopore sequencing would be if you already know what you're looking for, if you already have a model or a machine learning model or a framework which you have tested